2019, the FDA has just classified psilocybin as a breakthrough therapy for treatment-resistant depression. This classification helps to accelerate the process of clinical trials and the general approval process. Currently, there is a clinical trial in phase 3 of testing in order to establish psilocybin therapy as a possible treatment for depression. And in addition to treating NDD, it may be helpful for anxiety, those with cancer diagnoses, as well as addiction/abuse disorders. Psilocybin is the main active component of hallucinogenic mushrooms, characterized by feelings of euphoria and altered perception. It also contains a very low number of dangerous side effects found during usage. In preliminary studies conducted by Timothy Leary, participants reported the experience of disorientation, lethargy, giddiness, and euphoria. The range of responses doesn't cast doubt onto the potential of psilocybin, but more research is necessary to determine why negative experiences, although a minority, may occur. However, in the 1960s, it was classified as a Schedule I drug, which is the most heavily criminalized category, making its availability very low for medical and scientific use, until recently. Okay, our resident BNFB scientist will go more in depth on the mechanisms, but as a basic overview, the active component in psilocybin is a serotonergic agonist that specifically binds to your 5-HT2A serotonin receptors, and not only increases general activity, but also increases connectivity into sort of an organized chaos. These effects were not discovered recently. Originally referred to as Teonanacatl, or God's flesh, these sacred mushrooms originated in Mesoamerica and have been used in both therapeutic and spiritual settings for many years. Now we will transition to our very qualified scientist, lab researcher, to tell us more about the mechanisms of this substance.
When psilocybin was first introduced into experiments in the late 1950s, scientists couldn't detect a noticeable difference in mice and rats who had psilocybin in their system. A scientist then tried psilocybin in order to discover the hallucinogenic effects. Why does psilocybin not have any effects on mice and rats? We know in its purest form that it's a white crystal and that it's soluble in water, and it was known as a magic mushroom in Mexico. But why is there no effects on mice and rats? Maybe I'll just have to take it myself. Whoa, I'm definitely feeling a sense of euphoria, which is probably because currently in my body, psilocybin is being dephosphorylated into psilocin, which is the main active metabolite of psilocybin. Psilocin is then distributed to all parts of the body and binds to many receptors, including serotonin and dopamine. It binds to receptors through a cation-pi interaction, which occurs part of the negatively charged anion of psilocin and the electron-dense tryptophan. This reaction then results in the binding of receptors, which is why I currently feel a sense of euphoria. After this was discovered, psilocybin was classified as a Schedule I drug, which is why it was not used in research, up until recently.
Thank you. Potential benefits may outweigh the potential risks. In 2 studies conducted in 2016, psilocybin was linked to long-term improvement in mood in 80% of participants. Similar trends were noted to those addicted to smoking. But to date, it remains a Schedule I drug, labeling it as a high risk for abuse and no accepted medical use, even though the data does not support this classification.
Have you been struggling with treatment-resistant depression? Maybe psilocybin is right for you. If you have tried more than 2 medications along with psychotherapy that have not successfully relieved you of major depressive disorder, you qualify for this new approach. This substance activates serotonin receptors, especially in the prefrontal cortex, affecting mood and perception. In conjunction with psychotherapy, this treatment can lead to a new outlook on life altogether. In one study, psilocybin decreased depressive symptoms in all 19 participants after just 1 week. In this study, functional magnetic resonance imaging showed decreased cerebral blood flow in the amygdala, which has a correlation with decreased depressive symptoms. Even 5 weeks after a 2-dose treatment, benefits remain in the increased resting state functional connectivity. Almost half of the participants said they continued to notice tangible positive effects of the psilocybin. Ask your physician if psilocybin is right for you.
Side effects may include mood changes, anxiety, nausea, paranoia, euphoria, hallucinations, out-of-body experiences, drowsiness, heightened sensitivity, and confusion. Ask your doctor today.
So far, some media coverage has definitely sensationalized the potentially positive effects of this therapy, as this is not going to affect all people positively. The setting in which one uses psilocybin plays a large and important role in the response, so while there aren't any detrimental effects from the drug itself, for example, it is not linked to addiction, a bad trip is possible and may cause a cascade of negative effects. There are several clinical trials using psilocybin for the treatment of MDD, anxiety, OCD, body dysmorphia and eating disorders, Alzheimer's, cognitive impairment, alcohol, and other substance use disorders, and even migraines. Clinical trials in people with these conditions vastly outnumber the amount in healthy participants. It will likely be several years before we thoroughly identify relevant mechanisms and how to mitigate potentially negative effects and determine long-term use risks. Researchers have yet to establish the mechanistic relationship between a subjective psychedelic experience, psilocin levels, and 5-HT2AR occupancy in the brain. However, in a groundbreaking study published in September of 2020, researchers used advanced methodology called cryo-electron microscopy to create a 3D image of how psychedelic compounds bind to receptors. This research establishes the possibility of eliminating the hallucinogenic properties while maintaining strong therapeutic effects. The interaction between the 5-HT2A receptor and GQ residues, a family of heterotrymic G proteins, are the cornerstone to forming the selective engagements of the 5-HT2A receptor. In the next couple of years, as methodology advances, we will likely be able to identify more specific interactions between the amino acids and the serotonin receptor and the psilocybin complex, and allow us to further understand the chemical properties. Thank you for tuning in to BNFB News. Please email us if you have any questions.